Awesome. So with that, Alex will remain on screen because we will move into his next presentation, which is the application of CEDAR and the data within. So Alex will now present CEDAR, the prostate cancer meta-analysis. Thank you.
Okay. So let me put it on presentation mode. And so in this next presentation, so remember the presentation that you just heard was supposed to be given by Zainab. And so now I come in and give you a specific example of a meta-analysis of the defined epitopes from prostate-specific antigens. So what does it mean? As we discussed, the cancer epitope database, CEDAR, this new initiative, and these are two recent publications that substantiates these bullets. And as we were developing the database, as I was explaining in the previous talk, we also started curating prostate and neoepitopes. And so the choice was by design, in the sense that prostate cancer antigens are a set of very well-defined antigens associated with prostate tissue and can be targeted by prostate responses. And in general, also prostate cancers are cold tumors and were not associated with a high level of mutations. So that was an attractive feature of having a well-defined set of antigens and not so many mutations in the context of instead neoepitopes, which are as different from prostate antigen as possible, in the sense that the neoepitopes can pretty much originate from any antigens, and also by definition are mutations. So what I'm going to do today is present a meta-analysis of data related to prostate antigens, and the point of that is to give you a flavor of what are the capabilities of CEDAR at this point. It's good to also solicit feedback in the sense that one can talk about what is the blueprint for the database, but concrete examples of what can be done are a lot more informative, and in our experience can trigger much more detailed feedback and in-depth discussion. And certainly we have used this strategy a lot in the context of the IEDB, where we routinely have done meta-analysis, for example, of TB epitopes or SARS epitopes or dengue epitopes, and engaging the community has been really very helpful. We realized things that we had not realized, whether we had missed important papers or there were some peculiarities about that particular space of antigens and epitopes that needed to be taken into account, because that's what people working in that space cared about. So it really helps us do a better job. At the same point, to really look at the whole totality of the epitope data related to a particular application, it's important. And because you're looking at the forest rather than the trees. And so this identifies strengths and knowledge gaps related to, in this particular case, to prostate antigens. So here is recapping the process. As always, we start for curation from a text classifier. We identified potentially relevant cancer-related papers, and we had of a total over 10,000 different cancer-related papers. Of those, about 400 were classified as related to prostate antigens, and we're done essentially. And as we did in our successive layers of curation review, we found out that in reality, about 165 that contained relevant epitope curatable information. At the same time, we wanted to make sure that this process of the classifier and identified papers was working correctly. And so we did some independent literature search to make sure that our definition in the classifier of prostate antigen was not missing some important prostate antigen. We did that, and we were pleased to see that, yes, the classifier seems to be working fine. So in the following slides, I will talk to you about the data related to 8 prostate-specific antigens. We'll show you in the next slides what they are. And I want to make sure that I'm clear on the fact that we are looking at prostate-specific antigens. So these are antigens that are only expressed in the prostate and in prostate cancers. So, for example, you may have that a prostate cancer may express MAGE or some other receptor or whatever. But if that is not a prostate-specific antigens, that it's a different set of antigens. For example, MAGE will be curated separately. So it is a cancer-specific antigen, but it's not a prostate-specific antigen. So very important to appreciate that in the scope of this analysis. So we can query, as I was showing you earlier in the previous presentation, we can enter the information. And here, if we look at antigens, all process-specific PSA and positive assays, and no B-cell, no MHC, you will find 47 T-cell assays, for example, in this particular example. And as in the case of the IEDB, you will get, clicking on the different banners on top, different views of the data, and the assays here will tell you what assay these different epitopes were tested in, the restriction, and the outcome of a particular assay. So this is the data for the 8 main prostate antigens. So it's for those of you that may be familiar with the prostate antigen literature, it's a PAP, PSA, PSM, prostate, and PSCA, and so forth, but are really the most well-known and well-characterized prostate antigens. And you see here different columns provide different information. Here is the different references for this 8 main. There are a few minor other antigens are for the sake of this analysis. I'm just going to focus on the these main references here, and as you can see, PAP and PSA and PSMA are really the heavy hitters, and this is a protein gland, and I'll get back to that. And this is the number of epitopes. So overall, grand total about 300 epitopes been defined. one interesting way to look at the data is to say how many epitopes have been defined in humans versus mouse. And you see that for PAP and PSA, it's about the same. PSMA, it's more human than mouse. And for these other antigens, I mean, CIP is about the same, but for prostate and PSA, it's very much only human. Also, if you look at the ratio of B cell to T cell, PAP has a lot more T cell than B cell. PSA is similar as is PSMA, and conversely, prostate is very heavily skewed towards more B cell epitope information and so forth. So there are interesting trends and different biases, if you wish, in what information is available. And some this analysis also points out specific gaps. So for example, the scarcity of T cell epitopes for prostate, or conversely, the scarcity of B cell epitopes for PAP and so forth. Going back to the number of epitopes, what we did was to look at the correlation between number of epitopes and either number of publications or protein length. And why did we do that? What is normally used in well-characterized systems, like, I don't know, for example, dengue or hepatitis B, where there is a lot of data, it's common to see that the number of epitopes is correlated with how big an antigen is. And this is basically because a big antigen has a lot of potential different peptides. And so you had just basically more chances to win the lottery, to be a good epitope. That is not the case. There is no correlation between protein length and number of epitopes in the case of the prostate antigens. What is there, though, is instead a correlation between the number of publications and the epitopes. And so this is really a hallmark of the fact that this field is not sufficiently mined. In other words, every new publication finds new epitopes because there is a lot of uncovered territory and a lot of epitopes that are waiting to be reported and discovered. So as highlighted throughout this workshop, we've curated the specific assays linked to particular epitopes. And so here you see the data, but for the 4 main antigens and categorized by T cell assays. And you see that there is a fair number of assays being described. The number of epitopes for which there is a assigned class 1 and class 2 restriction varies again, and in general is a minority of the total assays. A minority of a total T cell assay have a defined restriction, and this becomes even more apparent when you take notice of how many of the T cell assays and antigen epitope assay combination are associated with tetrameric reagents, tetramers, and very, very few you see here. And this is a major knowledge gap that is in terms of characterization. You see there are few assays, and in fact, the number of epitopes for which there are tetramers, it's really a handful. And in terms of MHC ligand data, again, there is some data, not a lot of data, and also particularly deficient in terms of ligand with well-characterized restrictions. Another point, as is often the case, the restriction data is heavily skewed towards the HLA molecules that are, particularly in the case of class 1, most frequent in particular ethnic groups. And HLA-A201, it's a very well-studied allele, very frequent in Caucasians. A2402 is also a frequent allele, particularly in Asian ethnicities. And that's about it. You see that there's very little additional epitopes that have been defined. And you see in the case of HLA, particularly DR beta-1, there's a lot more spread of alleles, few epitopes though, and notably no DP, no DQ restricted epitopes. So major knowledge gaps here at the level of HLA restrictions. Another thing that we do is record what type of cytokine assays is detected in the epitope specific data that is curated in the IEDB. And you can see that the vast majority of the records relate to interferon gamma, which is a pro-inflammatory and clearly important cytokine. However, there are other cytokines that are important. TNF or, for example, IL-10, crucial cytokine linked perhaps to a more suppressive environment, such as can be found in the tumor microenvironment, or granzyme B that is relevant for cytotoxicity and so forth. So on one hand, good news, there's a lot of interferon gamma data being reported, relatively speaking, but major knowledge gap in terms of really assessing the multi-specificity of T-cell responses. B-cell epitopes are a little better in the sense that there's a little bit more balance equilibrium between monoclonal and polyclonal responses, and different assays being utilized. Again, if you look at, for example, how many epitopes are associated with the crystal structure, there is very, very little. One assay that is in particular very important in the context of cancer epitopes and cancer antigen is the type of assay where we observe recognition of cancer cells or killing of target cells. And there are relatively few instances in which this crucial assay has been reported. And also in terms of how to extract this data, it's currently not immediately easy to extract from the CEDAR database, but it's totally possible if one searches for tumor cells and antigen and exports the data and does a search for cytotoxicity. Equally sparse data is available in terms of linking reactivity to a particular prostate antigen epitope and an in vivo effect, decreased disease, changes in tumor burden or survival. And this is largely a reflection of the fact that there is a scarcity of mouse models for prostate antigens, and of also differences in anatomy between mouse and human prostate. But still, again, it's really a handful. In most cases, you can count the instances in which a particular epitope has been linked to an in vivo activity in the finger of one hand. Another knowledge gap that the prostate meta-analysis uncovered is the scarcity of defined TCR and BCRs that recognize specific epitopes derived from a prostate antigen. The data is only from PSA, and again, 1 or 3 receptors, which is very, very limited. So these are the conclusions for T-cell assays, mostly interferon. PSA is the only protein that has multi-specific records. Very few tetramers available in the scientific community. Some epitopes identified in ligand elution assays, but again, relatively few. A bit more diverse spectrum of B-cell assays. And limited data regarding tumor killing and in vivo effects. And lastly, 4 TCR and BCR total data relating to those receptors engaging epitopes derived from these prostate-specific antigens. And just as a point of comparison, for COVID, there is almost 100,000 TCRs that are available. Another component of the granularity of the data that is available through CEDAR is the classification for the records of what type of tumor and cancer is associated with the epitope recognition. And you can see here that the vast majority of records are related to prostate adenocarcinoma. And quite a few are prostate cancer. And this is basically a reflection of the fact that the author of the paper may not really discuss or define with more precision what type of prostate cancer, and it's quite likely these are indeed mostly adenocarcinoma, just that it's not defined. There are a few records related to the castration-resisted prostate carcinoma, which is a later stage and more metastatic, more serious type of prostate cancer, which I think it's probably less studied, but it would also be interesting to get additional data related to that. One thing that we have seen is that at this point, we can improve the granularity of what is curated, obviously provided that that information is discussed in the paper. Particularly, we have cancer type, but not staging. And so this is an ongoing process. Finally, last point is applying the immunobrowser to the prostate antigens. And this was one of the reasons why we wanted to look at the prostate antigens, because we knew that these are well-defined antigens for which to which we could apply the immunobrowser. So here I'm showing you an example for PSA with class 1 on top, class 2 in the middle, and B cell on the bottom. And you see that there are discrete regions that pop out. And in some cases, there is a overlap for example, between class 1 and class 2 in this region, or conversely, this region, there is a strong overlap between class 2 and B cells. So this is interesting in the sense, if one wanted to focus on one particular domain or part of PSA, one could zoom in on, for example, this region here is both targeted by class 2 and B cells, so it would be a fragment that would be of interest because you would have in the same fragment the capacity of eliciting a class 2 restricted T cell response, helping for the development of the antibodies. And conversely, this might be an opportunity to induce both a CD4 and CD8 T cell response, but less so in terms of a B cell response. And we've done this analysis for all other prostate antigens. And interestingly, there is a very significant correlation between different T cell activity for PSA. It is a relatively small protein. But for the larger proteins, such as PSM, for example, the correlation breaks down, and that is relevant because those proteins are bigger, and so one would have to consider in the context of perhaps a more refined or epitope inducing strategies, the influence of having one activity and not the other. Overall conclusion here, essentially we're done with the curation of prostate antigen epitopes. There's a lot of data out there, but also the analysis reveal very significant knowledge gaps. Lots of still needs to be done. Limited number with receptors, with tetramers, in vivo and tumor recognition assays, and so forth. And we also identified some areas for potential improvement of CEDAR, and the immunobrowser finally revealed immunodominant regions for the different main adaptive arms of immune response, and sometimes they correlate, sometimes they do not. And with that, I will stop and go back to the screen here.
Thank you so much, Alex, and absolutely perfect timing with that. As we give you a little bit of time to look at some of the questions in the chat, Bjoern has joined us, and he'll be showing you a brief demonstration of the CEDAR database now, and then we'll move into the Q&A. So thanks, Bjoern.
Yeah, so Alex, everything that Alex was presenting obviously is based on the data that is in CEDAR right now. And I'm just gonna give like a quick live demo. This is the starting page again, and now you can select things like I want to see human epitopes, T cell epitopes specifically that are class 1 restricted. So once you search for that, this is the answer that you're getting. Again, you have your different tabs for the different types of data. When you look at antigen here specifically, so you have the epitopes that are mapped to antigens, and then you see here actually the most frequently characterized antigens are viral antigens that are associated with cancer. If you're saying I don't care about, say, herpes viruses, etc., you can exclude the viral antigens and redo your search. Now you again like have the antigens here. You see now the most common ones are MART1, prostate-specific antigen, and again, like Alex was pointing out, we have done selective curation so far, focusing on prostate. So you see a lot of prostate-specific antigens in the top of the results. And as Alex was saying, for example, if you now specifically care about assays that are like cytotoxic responses, you can go down to your assay type, select specifically epitopes that have been involved in cytotoxicity, do that search again, and now you have your top results here, epitopes, with the top one being cancer testis antigen. So again, this is very much built on the IEDB system. In the background, the data is being captured like in the IEDB, but CEDAR is supposed to give us targeted query capabilities that highlight obviously the features of cancers. So then specifically, obviously mutation is one of the key things in cancer. So we have the ability to search for just neoantigens. You can do that. And now you'll only have mutated epitopes. And that is going to take me to one of the Q&A questions that came. Do we capture, or are we planning to capture, somatic mutations versus germline mutations? So germline mutations are things that you're born with that might predispose you to cancer, like certain BRCA, breast cancer antigen mutations, versus the somatic ones that are originating with the cancer itself. We are currently not capturing that difference. It's actually very hard sometimes to know if in the paper they didn't do a thorough study of that. But it's a good example for something that, because in the IEDB we didn't used to capture this, we hadn't started CEDAR with this in mind, but we will capture these kinds of distinctions going forward. And with that, I hope Alex had some time to look at the questions, and he can take the first one right now.
So yes, so I'm going to answer Amy Rosenberg. She asked, if these prostate antigens are present on normal tissues, is there no tolerance to them? Would you not have to break such tolerance to generate responses when they are expressed on tumors? Yes, so I mean, that is a general, it's a general consideration, and that was portrayed as one of the main advantages of neoepitopes, because neoepitopes are not present on normal tissues by definition associated with mutations, so you don't have to break tolerance. Now, the flip side is that the neoepitope approach forces you to have personalized cancer vaccination. So, in fact, historically, all the original first set of data was done with antigens that were not mutated. So prostate antigens are some of those, and the same applies to MAGE or to, and so people have tried to break tolerance. And when you do break tolerance, which is actually is not that hard, in a way, you can induce fairly readily responses against self components, particularly with adjuvants and so forth. But then the question becomes, are you going to have off-target problems? Because after breaking tolerance, conceivably the T cells will go on and attack also the normal tissue in which that is expressed. And so again, historically, people have focused on antigens that you can live without. So you can live without a prostate. And again, some of the same story applies to a certain degree to ovarian antigens or breast cancer associated antigens, because of the fact that it's painful, it's very bad, but you can have surgical removal of certain things, and or antigens that are overexpressed in tumors, and there the notion is that basically you will have that the T cell will only react to, or mostly react against the tumor. Or in the case of MAGE or melanoma antigens, it's clear that successful vaccination is associated with vitiligo. So where you also eliminate melanoma pigment producing cells from the skin and so forth. Bjoern?
Yeah, thanks, Alex. So I'll do the question from Franz Danny, which I kind of answered already. Will we be able to different mutation type associated with the cancer neoantigen, notably somatic versus germline? So as I showed you right now, we're not doing that, but we are planning on capturing the type of mutation Right now, what we are capturing is essentially the mutated sequence and the wild type sequence, which in the case of a single nucleotide variant, which leads to a single amino acid change, is straightforward. But you also have things like frame shift mutations or so, where it's actually very unclear what is the unmutated sequence belonging to a certain frame shift or so. So again, that is going to be coming in the future. Right now, no. Alex?
I will answer the question from Mauricio Calvo Calle. Immunodominant regions in cancer could be position prone to mutate. Yeah, I mean, that again, it's a great comment, and that is something that has been much debated over the years. In the specific case of prostate antigens, these are cold tumors in general, and you don't see a lot of mutations. And why is that? That it's open to debate. I don't have a good explanation, but in general, it is possible that some of the mutations that accumulate in tumors may be just originating because of T-cell pressure, but I think that the general impression is that the mutations that accumulate in tumors are not driven by T-cell pressure very much, and maybe related to other either just mutations because as a reflection of the dysregulated growth of a cancer, or having mutations involved with advantage growth of the cancer and so forth. So I didn't give you a very definitive question or answer, but I don't think that there is a very definitive answer in the field, period.
So then I'll take the next one. This is from Gerald. Are CEDAR epitopes a subset of IEDB epitopes, or what happens if you search for a certain sequence in the main IEDB site? Yes, they are a subset. So essentially the way our curation works, everything goes into the IEDB. So every bit of data that you see in CEDAR, you will also be able to see in the IEDB, but maybe a little bit differently presented, because obviously in the IEDB, we have a broader scope. So the way actually something ends up in CEDAR is on a per antigen level. So we are identifying which antigens are studied in a reference where a person has cancer, and you have an immune response to epitopes from that antigen due to the cancer that they have. And then we consider that a cancer antigen, and will include every paper in the IEDB that has epitopes from that antigen, and include that in CEDAR. So specifically, if you have something like somebody studying MHC binding, there's obviously no cancer involved. You just have like a peptide that is you're measuring affinity, but obviously that is important, and you want to know about that when you're looking at that antigen in detail. So then essentially a reference and all its data is going to be included in CEDAR if there's at least one epitope studied that comes from an antigen that is cancer associated. And this has worked quite well so far. We'll see if this classification holds up going forward, but so far so good. Alex?
Yes, Mauricio, again, lots of good comment. Thank you, Mauricio. Do you plan to include proteomics or RNA data to correlate expression with T-cell epitope response? And Bjoern, keep me honest here if I don't answer correctly, but the answer shortly is no. Not because it's not interesting. In fact, proteomics data and RNA, we have been generating analysis which show that really combining knowledge about, for example, the predicted MHC binding capacity or the mutation or whatever you, with expression levels really improves the capacity to predict T-cell epitopes. We do not include that currently in CEDAR or IEDB because also data sets could be different, depends on expression where, in a cancer cell, in a normal cell, in an infected cell, and so forth. So it becomes its own life, so to speak. But certainly, I think conceptually to combine that is of great interest. And I don't know if Bjoern, you want to add anything to that.
Yeah, so Alex is completely right. I think we consider this a little bit for tool development. These are really interesting questions. Obviously, there are lots of databases out there that do gene expression and proteomics data very well and have done that for decades. So our approach is going to be more that as we are trying to do this tool development, to essentially see for which of the references that we're curating is there a data set deposited that has gene expression or proteomics data and these other references, and then try to compile for these tool development purposes data sets where these things are available. So this will not make it in the CEDAR database proper, at least that's not our plan, but we'll have like curated data sets for tool development purposes where that data is available, because you really want to have the whole thing. Just including, say, one expression data point is kind of meaningless. You'll always want to see the overall gene expression data sets this is based on, and that would be a huge amount of data that we can't possibly store as a side project. I think that's it.
That's it.